glycogen, glycolysis. Glycogen, yes. Why do we need glycogen?
For the production of ATP.
To make ATP, actually we need glucose.
Yeah.
So the glycogen actually glycogen is like this ball of glucose molecules that are all strung together. Very simple. So we can break down glycogen and produce more glucose, and then we need glucose for ATP. Exactly. What else do we know about nutrition and metabolism?
So, not everyone's metabolism is the same.
Not everybody's metabolism operates exactly the same. No, some have a much more rapid metabolism, if you don't think of it that way. Yeah. More efficient, I don't know. So it just, nobody operates exactly the same. Yeah, what else? What are we metabolizing?
Fats.
Fats, proteins, carbohydrates, exactly. And each one of them has, of course, an important part of our structure. Yeah. Well, so important that we metabolize fats, carbohydrates, and proteins, but we also have to remember that metabolism and nutrition involve minerals and vitamins. We'll spend some time talking about them very shortly. We are going to obviously discuss how we get the breakdown going, which is why I say watch that guy, because if you haven't watched him, he's going to help you understand what's happening here. Watch him. They're rarely easy to watch, they're very easy to understand, and they are very succinct. So make sure that you watch this guy. We're going to talk about how we get our carbohydrates, lipids, and proteins down to their base components, to their monomers. And we've actually already looked at this, so we know that our carbohydrate chemical digestion begins in the mouth because we produce amylase. Which salivary gland produces amylase?
Parotid.
The parotid, very good. So we have salivary amylase that helps us start the process of hydrolyzing carbohydrates in the mouth. Not much happens in the mouth with regard to lipids, even though we make lipase via the lingual lipase glands, lingual lipase. That lipase has to be activated when it hits the stomach and gets activated with HCl. Nothing happens to proteins except for mechanical digestion of breaking them down just from large chunks to smaller pieces. That's pretty much it with all of these, more or less in the mouth mechanically. Nothing happens to carbohydrates in the stomach. We have a little bit of gastric lipase and lingual lipase get activated in the stomach, so a little bit of exposure to some enzymes for the lipids, but really nothing significant. And then, of course, the biggie here is the production of pepsin, which then begins the process of hydrolyzing our complex proteins to polypeptide chains, is really what happens. So we go from the tertiary structure to the secondary structure. Then we get to the duodenum and the small intestine, and we have the pancreas producing amylase, the pancreas producing lipase, the pancreas producing trypsinogen, and all the others, and then the trypsin that further produces these additional peptase or proteases. The carbohydrates at this point turned into disaccharides, sucrose, glucose, fructose, that sort of thing. Glucose is a monosaccharide, but sucrose and fructose and galactose, disaccharides. We produce the peptides in the stomach from the exposure to these pancreatic proteases. And then in the small intestine, we produce our monosaccharides, we produce our fatty acids, and we produce our amino acids in the small intestine. And we might remember this from when we talked about carbohydrate absorption in our digestive system, that we have on the luminal surface, glucose is taken in by sodium. It's called a symport. So we move sodium in from the outside, carries glucose with it. We move galactose in from the outside, carrying sodium with it. And then that sodium is returned to the outside via the sodium-potassium pump. And I'll get to that potassium in a moment. And why, I don't know. Fructose gets its own facilitated fusion protein.
Special.
The fructose, pardon me?
Special.
Special, yeah. That fructose and the galactose, they can be converted to glucose either here or in the liver. And we also have this solvent drag through the tight junctions. So they say tight junctions are keeping things from going through, but not everything. So water can drag glucose and some other solutes through these tight junctions. And all of that material is then from the core of the villus sent to the circulatory system. The potassium is kind of interesting, so I was asking Dr. Hill about it, and she said, well, this is what I show them. You know, we have these leak proteins all over the body. All of our cells have little leak proteins, and so the potassium is allowed to leak out, and the sodium is allowed to leak in, and that's why we have the sodium-potassium pump, to continually establish that balance between sodium and potassium. And so while we are moving potassium into the cells, we know that we are also losing potassium through these leak channels, and so we have the sodium-potassium pump pushing that sodium back in. Because look at all the leak channels. All those are ways that potassium is just going down its concentration gradient, just trying to establish its equal on both sides. And we all remember this. This is the simplest, I think, of all the mechanisms by which we move materials. We produce the amino acids. The amino acids are taken in via the symport along with sodium. The amino acids are moved to the circulatory system. The sodium is then removed out of the cell and back into the exterior along with potassium back in. So even all these intestinal cells, right? Potassium is leaking out, and we're always replacing it by the presence of sodium inside the cell and moving it via sodium-potassium pumps. Lipid absorption. So we are really aware now of how we take big globs of fat, we emulsify those globs of fat by the presence of bile. The bile salts and the lecithin form a little coating around these little globules. These little globules are called micelles. And the micelles are important because that's how we're getting vitamin A, D, E, and K from the intestinal lumen into the circulatory system. And we'll see why we need them in a little while. So these micelles have obviously allowed lipase to come in and hydrolyze the triglycerides, producing fatty acids and glycerol. And those micelles then just meld with the plasma membrane of our enterocytes, and the fatty acids and the glycerol are delivered to the inside of the enterocyte, and the enterocytes put them back together. And they coat them with a protein, and now we call them chylomicrons. And the chylomicrons cannot be absorbed by the circulatory system. By these capillaries, the endothelium are held together too tightly. So we depend on the lacteals. Remember the lacteals? The lacteals are blind sac, right, up into the villus, little blind sac lymphatic vessel with the gaping holes. And all of those nutrients that can't get through the circulatory system go through the lacteals. But in this case, it's these chylomicrons. Those chylomicrons then are going to be carried back to the circulation and then delivered via the thoracic duct to the circulation, and then they are carried in the circulation. And we'll see what happens with them in a moment. We have the nervous system involved in this whole process. Great, you get your food. You are delivered that food in part because you have a hormone called ghrelin that is going to tell you to eat. So back to the parietal cells. They produce hydrochloric acid, but they're also producing intrinsic factor, which was important for blood production, for erythropoiesis. And they also produce this appetite stimulant called ghrelin. Well, that makes total sense, doesn't it? If you're producing HCl and you're producing all these gastric juices because your cephalic phase is telling you, then it's kind of nice to have a hormone that says, tell you to eat. Another part of the cephalic phase is this ghrelin is telling you to eat. Other things that happen is you produce this protein called peptide YY, And the peptide YY, or PYY, is going to tell you to stop eating. So it is an appetite suppressant. It tells you you're full. You have cholecystokinin. Oh, we've already heard about cholecystokinin, where from those enteroendocrine cells. And cholecystokinin is going to stimulate the secretion of bile and stimulate the secretion of those gastric juices. And then it's going to tell you you're full. Why? Because you've got food in processing. You've got food in the pipeline already. So you don't need to add any more. So these 3 short-term hormones are going to help regulate our digestive process. And then we have some long-term hormones that help us digest or process our foods. And one of them is called leptin. And leptin is actually produced by adipocytes. And leptin also is an appetite suppressant, so it increases satiety, it tells you you're full, which makes total sense. The more adipocytes, the bigger your adipocytes, the more leptin. Like beautiful. However, they think that there are people who are missing the receptors for leptin. And they also think that, I don't know if you know this, but all the retrievers, the Labrador retrievers, the golden retrievers, all the retrievers have insatiable appetites. They just want to eat, eat, eat. And there's some talk about them missing the receptors for this hormone leptin. So they're always like ravenously hungry, always. Yeah. Then we have insulin, and you wouldn't think of insulin as an appetite suppressor, but it is. So obviously we know insulin is important for taking up glucose. I didn't know about the amino acid thing, but yes, it participates in amino acid uptake, meaning we can add amino acids into our cells. It is involved with lipogenesis, making fats and making glycogen, and it increases satiety. And the way it increases satiety is by breaking through the blood-brain barrier and essentially telling your brain, hey, if I'm here, it means that there's plenty of glucose in the system, you don't need to eat anymore. Pretty much like that. All right, so I gave you this slide right here because this is all my notes from, like, why is that? What's this? What's that? So I thought I would share all that with you. Interestingly to know that the Y in peptide YY, and you'll see it again in other places, it stands for the amino acid tyrosine. Yeah, and that's where we're going to stop. So I'll see you on Wednesday.